And also, I can do
Okay, let's continue where we left off. Oh, wait a second. I have a cool device. Thanks, Sharu. Okay, let me see. Let me see how it fits. Ah, it works. Halfway through the semester and we figured it out. Okay, great. Okay, so you're enjoying this spectroscopy set of lectures, aren't you? Humor me. Okay. What is the difference between AA? Yes, right?
AA is absorption and AB is emission.
Very good. So that means in AA, we measure the absorption. In AES, we measure the emission of light. Okay. What's the source of excitation in in atomic? My brain is in proteins. In atomic absorption spectroscopies. Yes. Great.
AA as the plane.
Yes. So the ICP inductively coupled plasma. So the plasma is the source of atomization and excitation. We don't have a lamp. In atomic absorption, we have a lamp. And the flame is the source of atomization. And the lamp is the source of excitation. It's a common exam quiz to ask you what's the source of atomization and what's the source of excitation. Yes.
It's the answer is the lamp, because the flame is actually the source of the atomization.
Yes. So in the flame, we break down the bonds. So it's source of atomization, breaking the molecules into atoms, breaking the bonds in a molecule into atoms. And the excitation comes from the flame. Whereas in plasma atomic emission spectroscopy, both atomization and excitation comes from the plasma. The plasma breaks the bonds within a molecule to free the atoms, and it also excites the atoms. What is the type of energy absorbed? Type of energy absorbed. Do you remember what the energy types are? Yes, Steve? The electronic energy. So the types of energy, we have electronic, vibration, and rotation. So there is no vibration and rotation in atoms, and in atomic absorption or atomic emission, the atom is absorbing, so it's only the electronic energy. How do we get the minerals in solution? Meaning, you all did the labs. How did you bring those ash components, the elements or the minerals in the ash in a solution? We put concentrated acid. So you put your concentrated acid in the crucible, and did you heat it a little bit? Yeah, on a hot plate to enhance those minerals, to help them dissolve in the acid, and then you dilute it. Okay. All right. You also did calibration. So basically, we got standards that are 99.99 pure, You don't want any contamination, very limited contamination, and you prepared different concentration. You noticed in lab you had the calcium and the iron, and the iron had much lower concentration, the standard curve. What for the iron at much lower concentration than the standard curve for calcium. And this is because we wanted to capture the linearity of the concentration to absorption ratio. So we wanted to make sure that our quantification happens in the linear range. And we diluted the samples in most cases so that the absorption lies within the range of your calibration or your standards. In some cases, it was more concentrated than you wanted it to be. And look at this curve. When it is, the concentration is high, in a range that is not linear, you cannot have a good prediction because your equation of the line is a linear equation of the line. So if your concentration falls in a place where it's actually plateauing, you might, you don't get the correct answer. You might be overestimating or underestimating depending on the shape of the curve after that linear range. So you really want to be in that linear range. I thought I had a linear shape. So maybe it is between 0 and the first dash here where you want to focus different 5 to 6 different concentration in that range. Okay. So we sometimes have interference. And to avoid interference and wrong quantification, we try to work around it. So iron and zinc have very close lambda max. Do you remember what lambda max is? I'm always going to try to have you remember that term. Yes, Sophie? It is the lambda at maximum absorption. So it is the wavelength at which you get maximum absorption. And iron and zinc, that lambda max is very close. So for the determination of iron and zinc, we don't select lambda max. So we select slightly different lambda that is better for iron if we're measuring iron, or it's better for zinc if we're measuring zinc. We try to avoid lambda max because otherwise the zinc interference happens. If we're measuring iron, we might be as well measuring zinc with it. Background, incomplete atomization. So I think I talked about it with one of the groups today, is that if you have a big molecule that doesn't get atomized in the flame, it will cause scattering of light. Maybe I talked about it also in lecture last time. So sometimes in the case of calcium, if you don't break the calcium pyrophosphate by using lanthanum chloride, or not enough lanthanum chloride is used. So what could happen is either you get underestimation because your calcium precipitates and not reach the flame because of the big colloidal molecule, or if you had some lanthanum chloride and there is some big molecules that might not break completely in the flame. So you might have large molecules in the flame that don't get atomized, will scatter light. Or if your flame is relatively cold, so the flame is not hot enough to break the molecular bonds, then you will have those large molecules that they can scatter light, meaning less light will reach the detection or the detector, and then you get wrong high absorbance. Okay, so that is mostly causing overestimation. So light scattering causes overestimation. Transport interference. So if you have components in your reagents that you use that are contaminants, or they might scatter light themselves. So that's why we prepare the standard similar way that we prepare the samples, and we always run a blank, correct for those transport interferences. We talked about solute volatilization. If you have large calcium pyrophosphate, it won't get into the volatile state, it won't get to the flame, precipitates. The other one is ionization. This happens if the flame is too hot. So if the flame is too hot, you don't only break bonds, but you cause the atoms to ionize. And ionized atoms absorb light at a different wavelength. So this causes underestimation. So too hot of a flame will cause underestimation. Too cold of a flame will cause overestimation. Also, common question is list 2 to 3 reasons for overestimation, 2 to 3 reasons for underestimation when you are doing atomic absorption. So you can write this down and try to study accordingly. Any questions related to the lab, related to this? Again, here we cover theory, and in labs we cover practical, and a lot of the questions are based on lab that reinforces the theory. So that's why there's a lot of focus on lab. Yes.
So if it's too hot, it's overestimated.
If the flame is too hot, if the flame is too hot, your atoms becomes ions, and ions absorb at a different lambda max. So what do you think happens, Gray, when you have a different lambda max for absorption for ions? Andres, there you go. Okay. So if we want to compare atomic absorption in comparison to inductively coupled plasma, so there's a lot of advantages for inductively coupled plasma, especially for, you don't need lamps, you can measure simultaneously multiple elements. So this is all great, but it is expensive. So atomic absorption is to obtain that instrument is less money than ICP, but it depends how often you do mineral, and if it is really a major thing you do in the lab, like the soil sciences department. Atomic absorption is older technique, so we know it's quark, so we know the advantages and disadvantages of it. And another difference is limit of detection is much lower for ICP than atomic absorption. So that is another advantage of ICP, is detection at much lower concentration. So higher sensitivity, basically. All right, so this concludes atomic absorption. Any questions about that? So I'm going to move on to another section or topic within spectroscopy. So we are still talking about spectroscopy. We're not done absorption and emission of light, but here specifically we're going to talk about the region of the spectrum, which is the UV visible region. And a lot of analysis, especially quantitative analysis and food analysis, is done via spectroscopy in this range. Okay, and we'll give examples. Now, let's see what you know. I forgot that I had that. Okay. What do you know? Which of the following is true? Anybody wants to? Is there a brave soul? Nobody's gonna criticize you, Tyler. Let's see. E. Any thoughts? Rachel. So G, all of the above. Just B.
Yeah.
Yes, just B.
Yeah.
That just B. Molar extinction is unique, usually for different compounds with different functional group. It is the absorptivity. Molar extinction is also known as absorptivity. It is a constant, very much related to the compound that you're measuring. Every compound has its own unique absorptivity constant, and it's usually determined empirically by experimentation. So, and it is different at different wavelengths. So, in particular, so for the same molecule, absorptivity will be different, or the same compound, or species, it could be an atom too. So the same molecule will have different, or compound, or species, or atom, will have different absorptivity at different lambda. Keep that in mind. So when you change the wavelength, the absorptivity changes because there are different allowable energy levels at different wavelengths. Okay, so there is different allowable energy that could be absorbed by that atom or molecule at different wavelengths. So this absorptivity is really dependent not only on the molecule, compound, or atom, but also on the wavelength. Okay, so the first one is not constant, so A is not correct. And incident beam minus exiting beam is equal to the light absorbed is incorrect. Because, so let's think of a cuvette or a cube that you put in your spectrometer. And then there is the light that comes in. And then there is the light that goes out to the detector. So we call this incident beam, the light coming in, exit beam. So, but there is reflectance of light from the surface. There is some light that gets reflected. Also, there is some light that gets scattered. So the light that is exiting, subtracted from the light that is coming in, is not equivalent. Enough, because there are light that either get scattered, reflected, absorbed by an interference. So C is incorrect. So this leaves us with B. A highly concentrated sample might result in deviation from Beer's law. I'll tell you why. So let's say you have a lot of molecules. Let's talk about molecules rather than atoms. You have a lot of molecules in your system. So they can bump into each other. They're in too close of a proximity. And then the electrons on the surface of these atoms within a molecule can start to be exchanged between a little bit here, a little bit there, and that will deviate us from Beer's law. So that's why we always say, make sure you have the correct dilution when you do spectroscopy, and make sure your absorption is in the range of to 2.8 absorption unit, whatever. So if you have an absorption like 2, it's too high, and you have a lot of molecules interacting with each other and you lose linearity. So as a rule of thumb, make sure you have a diluted sample that when you measure absorption, it falls within this kind of absorption reading. Okay. What about this one? All of the above. Yes. All of the above. We'll talk about that. So in UV-vis spectroscopy, we measure either absorbance or fluorescence. And it could be qualitative or quantitative detection. Oftentimes, we do qualitative where we just want to detect presence of something, or we want to identify a compound. We don't want to quantitate. But in many cases, however, we want to quantitate. And we rely sometimes on the inherent signal. So meaning the compound or the atom we're measuring can absorb light or can absorb light intensely. Sometimes we're measuring components like glucose, for example, cannot absorb light. So it does not absorb light, it does not fluoresce. So we need to do chemically induced modification to produce a chromophore, to produce a compound in the reaction that will absorb light, and either we measure absorption or we measure fluorescence. So that's what is meant by inherent signal. The compound itself can absorb light and sometimes can fluoresce. Or we have to make modifications to generate a product that will absorb light or will fluoresce. We have many examples for using of UV-vis spectroscopy, quantification of macro compounds, large compounds like carbohydrate. We're going to do a lab on total carbohydrates assay by phenol sulfuric acid method. It's kind of a general method for total carbohydrate. And what we do, total carbohydrates don't absorb light. So what we have them do, we digest them into small monomers. We react them with a phenol. We get a colored compound, a chromophore. Then we measure absorption. We'll do this assay in the lab. And then another one is quantification of microcomponent. In this example, vitamin, thiamine, can fluoresce. So we can measure the fluorescence to determine thiamine, or to produce a molecule from thiamine that will fluoresce. We'll cover that in the vitamins chapter. Or we use spectroscopy, another example, in ELISA. Enzyme-linked immunoassays, we'll have a chapter on that. A big application for ELISA is to determine allergen. Of course, ELISA is used for many other components, but allergens is one major one that we use ELISA. Okay. So how do we do quantitative analysis? So the quantitative analysis is based on measurement of the amount of light absorbed by a reference beam as it passes through the sample solution. So this is our reference beam or incident beam passing through. The C is the concentration, the A is the absorptivity, and the B is the distance. The distance the light travels within your sample. And then you have the exiting beam or the transmitted beam. Transmitted meaning it's transmitted to the detector. So it goes from the sample to the detector. So it's transmitted. All right, so relative transmittance or absorption is correlated to the concentration. But there's a difference. Sometimes we measure percent transmittance, sometimes we measure absorbance. But if we want to quantitate, it is better to use absorption because it has linear relationship with the concentration. Transmittance does not have a linear relationship with the concentration. And it is the ratio of the transmitted beam over the incident beam. And you can calculate percent transmittance as such. So it's not used to determine concentration by using simple linear equation like the Beer's law. So absorbance has a linear relationship with the concentration, and it is the log of inverse T. So log of P0 over P. So that's that. And the Beer's law is applied where we relate absorption, absorptivity, distance traveled by the light, and concentration. Okay. So the concentration can be in any unit. Sometimes our concentration is simple as a percentage. Sometimes it is in nanograms per milliliter. Sometimes it's a molar concentration. Path is in centimeter usually, the distance traveled, and absorptivity, the A, is the unit is inverse centimeter and inverse concentration. Absorption doesn't have a unit. So when we put all the units, let's say the concentration is molarity and the distance is centimeter, absorptivity is the unit is inverse of these two, the concentration and the centimeter. They cancel each other, the units, so absorption doesn't have it. And absorptivity sometimes is written in an epsilon symbol. That's especially used when the concentration is in molarity. Another term for it, when it is in molarity, we call it molar absorptivity or molar extinction. The same. Molar absorptivity or molar extinction is this epsilon. So when the concentration is in molarity. All right. So what does absorptivity relate to? We talked at least about two of those. Yeah. A and B, we talked about that, yes. So it's the molecular properties, the functional groups that allow a species to absorb light, and the wavelength. It changes with the wavelength. Same compound will have different absorptivity at different wavelength. What about chemical environment, and what do we mean by chemical environment? Yes, Mitchell. Yeah, well, yes, if you have high concentration, you have electron exchange, also the pH. So the pH will impact the charge that you have, the ionization state that you have, or if you have salt in there, that will also cause interaction of salt with the surface of the molecule. So these will impact how this compound will absorb light. So that's what is meant by chemical absorptivity. So the answer is all of that. So sometimes you have a reference cell in the spectrophotometer. Who worked with a spectrophotometer before? Very good. Sierra, do you remember if you had a reference cell in there? Yes. What about who else raised their hand? Do you remember if there's a reference cell? Huh?
He's like,
Blank cell, that's the reference cell. So sometimes spectrophotometer have a place where you put a blank and another place your sample. And then the beam of light is split. One goes to the blank, to the reference cell, and one goes to the sample. And it kind of corrects for any absorption interference that comes from the matrix or the reagents you use for your sample. So sometimes the blank is just simple water because there's nothing else really with your sample. Sometimes your blank is actually everything you have used in preparation of the sample without the sample. So the reference cell corrects and gives you a corrected reading, like automatically. You don't have to worry about correcting the absorbance. Sometimes you don't have a reference cell built into the spectrophotometer. So what do you do when you do not have a reference cell? What does logic dictate? Say that again. Yes, exactly. You run the blank, you can zero, you can put the blank and zero the equipment, take the blank out and then read your sample. Or you can read the blank, take it out, write down the absorption of the blank, then read your sample, then subtract it on your Excel sheet, the sample minus the blank. So this way you're always correcting either by zeroing or subtracting. And if you have a built-in reference cell, the absorption comes out corrected for you. So the reason we do that is to correct for any, absorbing solution or whatever is in the solution that scatters the light, and also correcting for the reflectance and any interferences in general. Okay, so that's what the image is for. So in order for quantification, we need to make sure that we meet the requirements of Beer's law. We do not want to deviate from the law. So these are instances where we deviate from the law, and that is when we have high concentrations. You have interferences, your absorptivity is not regular or constant. So we make sure the concentration is within a range that doesn't cause for that high interference. Oftentimes, we make sure we are working in a buffered system, especially if we have components that are impacted by changes in the environment, dissociation and association, especially if we're working with proteins, for example. So we make sure that the chemical environment is constant or protected from having changes within the molecule themselves, reversible association, dissociation. Another thing is we want to make sure that our monochromator is working fine, and the slit, the opening that we have within the monochromator allows a singular lambda or lambdas with very, very close length. So we do not want to have polychromatic light because then you don't have that constant absorptivity. So these are the three deviations of Beer's law, which you might be asked about in a quiz or an exam. Okay. So there are several procedural considerations. One is with sample preparation. Sometimes when you prepare a sample, you look at it and it looks cloudy to you. What do you do when you have cloudiness? Some of you in the lab, what would you do? Huh? I can't hear you. What?
Filter.
Yes, you can filter. Another one is, yes, we centrifuge. Oftentimes, we centrifuge to precipitate particulate because these particles are going to increase your absorption by scattering of light. And these particles come from your sample oftentimes, so you cannot correct it with the blank. The blank will have other reagents, so you cannot always correct for that scattering of light when you use a blank. So you want to make sure that the sample is homogeneous, it's clarified through centrifugation. Filtering is good, but sometimes particles we cannot see or are smaller than our filter pores. So oftentimes we centrifuge. And depending on the particles, centrifugation speed will be selected. So we use the reference cells, obviously, and the blanks to correct for anything coming from reagents. And the other consideration is the cuvette. This is called the cuvette. So the cuvette that we use, sometimes we only have limited amount of sample. Like look at it here, it's probably much less than 1 ml, maybe 100 microliter. We want to make sure that we fill the cuvette to a point where the light passes through the sample, not above the sample. So when you position your cuvette or tube, it is positioned in such a way that the light is passing through, usually mostly in the middle of it. So you want to have enough solution in there. Otherwise, if the light is passing here and your solution is right there, we're not capturing it. So what do we do when we have small amount of samples? We just use different types of cuvette. Like this, for example, that gives us the height we need that is sufficient for the spectrophotometer we have where the light passes through the sample. Another important is the type of the cuvette or tube that we use. So if we want to measure in the UV range, we cannot use glass or plastic. They absorb in the UV range. So we use usually quartz or fused silica. Quartz is really best, but it's very expensive. If you have it in the lab, that's good. You can use it and reuse it. If you're measuring visible light, you can use glass and plastic. And the glass is reusable. You can wash these tubes. Plastic you throw away. In the lab, you will be using cuvettes that are plastic for the total carbohydrate assay. You will use them and you will throw them away. Okay, so it's very important that we select the appropriate wavelength. Often we use the lambda max. This is kind of the go-to, the wavelength at which we have maximum absorption. But sometimes there are interferences, other compounds that absorb at the same lambda max. So what we do, we go to the next one. So let's say this is our, if you remember the spectra, there's the lambda, is the absorption. Sometimes you have like kind of a hill, it's higher. But at this, let's say 254, 275 nanometers. Here at this lambda, I might get multiple compounds absorbing here. I don't want that. Although this is the highest sensitivity, then I'll go to the next lambda here. That this other compound doesn't absorb that, interfering compound. So I might get less sensitivity. I just make sure I have more concentration. So my sample will be more concentrated. So selection of the wavelength is very important to maximize sensitivity, but also to minimize interference. It's not on the slide. You can just also add maximize sensitivity and minimize interference. Make sure you are differentiating these two. And also you want to make sure that that slit of that allows the light to go through at a particular lambda, it's small enough that allows only that singular lambda or wavelength that are very, very close to each other, like maybe 255 and 255.5, you know, and things like that. So it might not be exactly that one wavelength. There might be a little bit other wavelengths passing through, but they're very, very narrow so that we can be as much as possible adhere with Beer's law. So that bandwidth is really important. As small as we can get it so that we have the correct absorption, because sometimes you see here, very narrow. Sometimes if the bandwidth, the band here is wide, we can get 313 and 314 potentially, then we see quite a bit of a drop in absorption. So we have to be very, very careful in selecting that slit. Calibration is very important for the instrument. So you will do that, or the TAs will show you when you have your spectrometer to work with in the lab. We calibrate zero transmittance. So we block the light and we tell the instrument this is zero transmittance. No light is coming through, no light is reaching the detector. And then we don't have a cuvette and open the light and we calibrate it. This is 100 transmittance. This is usually done to avoid noise, instrument noise, like absorbances when there is nothing in the sample. Oh, this is the figure I thought I had in the other one. Okay, so here we want to make sure that we have linear relationship between concentration and absorbance. And we want to make sure that our absorbance of the unknown falls in this linear relationship. So we select the concentration where we have a linear line. We do not want nonlinear because we cannot predict correctly the concentration when the curve is nonlinear using the equation of the lambda. Okay, so I have 5 minutes. I'm going to try to explain this. In this 5 minutes. I know that you probably want to be done, but finish this. Okay. All right. Sometimes your sample, no matter what you do, you filter, you centrifuge, you have interferences that you cannot correct for in a blank. So this method, we call it standard addition protocol. So please remember that term, standard addition protocol. So this is to correct for components within your sample, not with the reagent, that you couldn't correct for by centrifugation, filtering, or any other attempt to correct for those. So what we often do is this method. So let's say we want to prepare a standard curve with 5 concentrations. So I'm going to draw it so that you can visualize it. You can also draw it on your sheet or notebook. Sorry, I'm not a good drawer. All right, so what you do is you add a volume of sample, the same volume of sample to all of them. So the same volume of your sample that is unknown concentration. We call it volume of unknown, VU. Okay, let me call it VU so that you don't get confused. Volume of the unknown. but it's the same volume. So if you dilute, you have your sample, you dilute it to the correct concentration, and you have 100 ml of that sample. That's same concentration. What we do is we just, let's say I put 10 microliter here, 50 microliter, 100. No, wait a second. What am I doing? No, sorry. Scratch that. Same volume. I was saying same volume. That would be for the standard. So 10 microliter, 10 microliter, and so I'm putting 10 microliter of the unknown. Then what I was doing is volume of the standard. So I prepare my stock solution of the standard, stock solution of standard. And here I put, let's change it, 20 microliter. So 20 microliter, 40 microliter, 80, 100, and 120. So are you all following? So I put the same volume of my sample, the unknown, in all of the flasks. Same. And then I put 20 microliter from, or different volumes, from the stock solution of the standard. So different volumes. Then I make up to the same volume, let's say 50 milliliter volume, total volume. Or GT. So now everything is in total volume of 50 ml. So I'm building a standard curve in the sample matrix. So the sample matrix is present in all of them, and I'm building my standard curve to allow for that interference so that my standard curve takes into account the interfering compounds that come from the sample. I plot the volume of standard because that's the one that is changing with the absorbance, and then I get the equation of the line. And then I solve for the concentration of the unknown. So that's how we do it. You get an equation of the line and you just solve for the concentration. So you don't have to remember the different terms, but what I want you to remember is how we build standards in the same sample matrix. And that you would prepare a standard curve that will correct for any interference. Any questions? Right on time. Okay. I will see you Friday.